A very good morning to all of you. First thing first, if the audio and video both are working fine, please let me know. Great, wonderful. So, yesterday we had begun with the chapter on biotechnology and we had discussed various aspects of biotechnology, the various fields in which biotechnology is applied. And this is where we are going to begin from. We discussed about stem cells and what stem cells are and all the things related to the properties and the applications of stem cells. So now the next topic for us is DNA fingerprinting. Now, in case of DNA fingerprinting, understand first and foremost what exactly do we mean by DNA fingerprinting and how exactly do we see the applications of DNA fingerprinting. What are the ways in which DNA fingerprinting is used, and where do we see some of the issues also which are related to DNA fingerprinting. So, we'll discuss all that. Now, before we begin about what we mean by DNA fingerprinting, think of fingerprint. What is special about fingerprint? What is unique about fingerprint? Can you tell me first? Yeah, it is unique. The unique thing about fingerprint is that for everyone, the fingerprints are unique. Now, when we say that it is unique, means that everybody has a different fingerprint. Similarly, when it comes to the DNA, each and every person's DNA also is different. Even if, let's say, people who are related, people who are a family, or even people who are twins, even in twins, what we see is that there will be slight differences in their DNA. So that's why each and every person's DNA also is different, but there are certain things which are common if they are genetically related. So that is where DNA fingerprinting becomes crucial, and we see that there are various applications of DNA fingerprinting. So that's why it is about identifying complete or partial set of genetic information of a particular individual. Now, when it comes to DNA fingerprinting, we see that there are various different applications. So, for example, it can be used in identification of criminals. Now, if you look at the picture that we have here, if let's say there is a crime scene sample or there is a DNA sample that we have received from a crime scene, this is where, if let's say there are suspects or if we are looking for people, there might we can collect or we might be able to collect DNA samples from them. And when we look at the DNA samples collected versus the crime sample that we got, can you see that there, out of these three suspects that have been listed here, there is one DNA sample which matches the crime sample of the crime with the DNA sample of the crime scene? Yes, suspect number two. So as you can see that this and this are similar, this and this are similar, and even this and this are similar. So all these three are similar. So that's why this is where we would at least know about the presence of suspect number two, because here you can see that this is different, this is different, this is different, this is different. So all these are different. Only suspect number two is where the entire DNA sample matches, and hence this is where they would be able to identify the presence of this person in the crime scene. Similarly, paternity tests can be done. We see that to establish paternity or maternity, we see that DNA samples can become useful, because there are certain things which will always uh, there are certain things which will be common in the DNA if they are being if they are blood related. So those kind of things will be seen and they will be trying to be established, and hence we'll see that it can also help in establishing paternity. Similarly, in cases of unfortunate accidents, we see that there also DNA analysis can be done, and that also gives an idea about uh, the DNA, and hence identification of a person can be done with the help of the DNA samples that have been collected from these kind of unfortunate accidents. Then, in making effective drugs, if you remember, we discussed yesterday something called as pharmacogenomics. pharmacogenomics. Now, in case of pharmacogenomics, what we discussed is this is where we are looking at how the genetic makeup of an individual affects has an effect on drugs. So that's why any medicine that is being developed, if genetics are being taken into account, if we are thinking of what the genetics are and what the genetic makeup of a population is, then we can make more effective drugs accordingly. And that's why we see that in that sense also DNA fingerprinting helps, because it's about identification of the DNA and making effective drugs accordingly. So that's why it will also be very very helpful in making drugs which are more effective and are acting better and faster in general. Similarly, in the treatment of genetic vulnerability also, we see that genetics become very very important, and identification of certain genes here, they become very very crucial, because then in that sense we'll be able to identify the things beforehand, and we might be able to prevent certain diseases as well. So that's why in that sense we see that it can genetic vulnerabilities can also be identified beforehand, and they can also be prevented. So that's why this is also very very crucial part of the entire setup. Now, and this is again something that we discussed, that when we talked about genetic testing, if you remember, we talked about genetic testing. So this is what genetic testing is about. Now, it can also become effective in wildlife management. Now, in wildlife management, there are a couple of things that we use DNA fingerprinting for. First is identification of DNA and what are the reasons that is leading to the drop in population of a certain species. That if there is an animal species which is where we see a significant drop in population, what are the reasons? If there are reasons that can be identified, many of the times we see that DNA will give us clues. What are the environmental factors or maybe anthropogenic factors which are leading to dwindling population of any given species? So that's why it is very very helpful in wildlife management as well. So that's why when it comes to DNA fingerprinting, we understand that there are a lot of ways in which DNA fingerprinting can become useful. Now, although it is very useful, we see that there are certain limitations or concerns over DNA fingerprinting as well. Now, first concern, for example, it can lead to reference for designer babies. If you remember, we discussed about this concept of designer babies, where we are saying that we are trying to pick and choose the traits that we want in the child, and accordingly each and every decision is being taken, and the genes are being identified, and only those genes are supposed to be used. So which is again something which is not ideal, because then it leads to in a manner commercialization of genes as well, that there are certain genes which will always be in demand and there are other genes which will not be in demand. And uh, it's also, you can say, in a manner an unethical practice, because the changes that you are making, the changes can be permanent and changes can go on for generation. So there is another term here called as germline editing. Germline editing. Now, in case of germline editing, what we are saying is that the kind of genetic editing that we are doing, the kind of genetic modification that we are doing, it can become permanent. It can become permanent, it can become hereditary, because when you make germline editing is done in a manner that the changes are being done in the embryo. Now, if the changes are done in the embryonic stages in these cases, what we see is that this is where once, especially in the reproductive cells, so if the changes are made in the reproductive cells, then they can be inherited from generation to generation. So they'll be passed on from generation to generation, and that's why this can become dangerous, because in case of cloning, what we did was the changes were being made in the somatic cells. So when you make the changes in the somatic cells, or whatever you do in the somatic cells, somatic cell changes will only be restricted to an individual. It will go not go beyond that individual. But when it comes to the changes that we make in the germ line, that is when we make the changes in the reproductive cells, this is where the changes that we make can be inherited for generations. So that's why this is something that also becomes an unethical practice, because the changes that we are making in the embryos or the reproductive cells like sperm cells or the egg cells, they can be passed on to the future generations as well. So that's why this becomes a dangerous trend then, because we do not know in what direction something like this can be taken, and how exactly these things can take a very different turn. So that's why germline editing becomes very, very dangerous. So that's why germline editing is process of making changes in the human genome, reproductive cells, or in the embryos. These changes can be passed on to the future generations. These changes can be passed on to the future generations as well. So that's why germline editing can become dangerous. Now let's say that in your child you want a certain trait to be there in your child, that you want your child to be the greatest physician of physicist of the world. So then in that case you are just going to look for those certain genes, and you'll ensure that those genes are present in the embryonic stage itself, and hence that person after birth might grow up to become one of the greatest physicists of all time. So you do not know, I mean, you are making those changes permanent, you do not know what course in the future those changes can make. It's very difficult to even predict what can go wrong. So it's not that it's that simple that you'll just pick up a trait and you'll just insert that trait, and everything that you do um, will just go on forever. You do not know what kind of changes can happen. But germline editing can be useful in treatment of genetic diseases, where some kind of genetic disease remains in any individual. To check for that, to ensure that that can be taken away, that genetic vulnerability can be taken away, germline editing in that sense can be helpful. But here in this case it will be a dangerous trend. Similarly, the genetic privacy of an individual will be violated. What may happen is that the genetic information, once, let's say, if uh, everybody knows about the DNA of everyone, then the entire gene pool is visible to everyone. And then in that case what happens is that you might start looking at what are superior genes versus what are inferior genes, and probably going further every decision is being made on the basis of those genes. That even if, let's say, a college wants admissions of students, it might see whether or not they have those genes which are suitable for a certain course or not. Or let's say in the employment also, maybe companies start looking towards those genes, and also distinguish between who they should give jobs to or who they shouldn't give jobs to. And overall the entire societal fabric will start probably behaving in this manner, where we'll start distinguishing between what is a superior genes versus what is inferior gene. So that's why there are all these issues and there are all these possibilities, and we do not know what course it might take. So that's why this can be a dangerous trend. And then also information can be misused for commercial purposes. Now this is also possible that once the genetic information is available, let's say the entire information about genes is available, then in that case people uh, might also see a situation where a lot of lot of companies which are related to pharmaceuticals, they might start selling medical packages. That they might say that, okay, fine, we have done the analysis of all the genes that are there in your body, and based on the genes in your body, we see that there are chances of this and that disease developing in the future. So this is the right time that you start taking precautions against it, and we are offering you medical package to take care of that. Now, uh, when they advertise things like these, and when they uh, try to hunt down the insecurity of a person, this is where, especially in medical science, we see that in the field of medicine, people will always be afraid of something going wrong, or uh, let's say if they instill fear in the minds of people, then in that case people will probably go and buy that medical package. And that's why these kind of things can also be, these will be obviously unethical practices, but these things can happen, commercialization of all these things can happen, and we do not know how the things will turn up to. So that's why we need to understand and the importance of not getting these information misused. Similarly, it can also lead to creation of weapons of mass destruction, because in case of weapons of mass destruction, um, maybe understanding of the genes will also lead to understanding of the vulnerabilities in the genes. So for example, let's say a virus, and we have seen what happened with covid-19, that covid-19, whether it was made in a lab in China or not, we will never know that, because it's China, you will never have transparent information from there. So but there are reports, there are reports backed by the US also and internationally also, which are claiming that covid-19 virus might have been made in a lab. But whether it was made in a lab or not, we have seen what kind of devastation it has caused worldwide, and we understood how exactly uh, the entire world suffered. And not only that, we see a situation where our immune system did not have an answer, right? It did not have an answer, and it took a while for the immune system to respond. The people who had stronger immune system, they might have been able to survive. Unfortunately, a lot of us did not survive. So that's why, and this is a scenario that can be made possible in the future, that let's say somebody wants to make a weapon of mass destruction against India, then all they have to do is they have to look for certain viruses or certain pathogens which can attack the Indian genes. And Indian genes, there have been intermixing of the genes for more than 1,000 years now, correct? Because in the south we are peninsular, so we are uh, there are water bodies, and hence it's not easy to cross over to India. And in the north also you have Himalayas, Karakoram, and even the Thar, which in a manner isolates this entire part, correct? So that's why what then happens is that there will be an intermixing of the genes in these parts, and there will be a lot of common genes which will be found in these parts. So this is where we see that the things can become dangerous, because somebody might want to just identify those common genes and try to attack those genes and make a weapon of mass destruction against it with the help of a pathogen like a virus. So there are possibilities, and all these things actually are possible. And that's why, from the perspective of DNA fingerprinting, it becomes very crucial for us to understand uh, how the things can go wrong. It's not only about all the applications. Yes, it has a lot of applications, but at the same time there are a lot of issues also, and we need to recognize all these issues also. Then a topic that we discussed yesterday, that that is gene therapy. Now, in case of gene therapy, what we discuss is that this is where we are making the use of genes to treat a disease. So it could be about replacing a gene which is responsible for certain disease. Then it can be about inactivating or knocking out a gene that is functioning improperly, or introducing a new gene into the body. So as we have discussed, that it could be modification, replacement, addition, or removal. So in these cases, in these ways, we can use gene therapy. And the way gene therapy works is very similar to how recombinant DNA works. Recombinant DNA technology, if you remember what we discussed in agricultural applications, gene therapy also works in a very similar fashion. So that's why there are all these ways in which gene therapy can work. Now, in case of gene therapy, although we have found that gene therapy becomes useful, and there are a lot of ways in which gene therapy can be used, and there are a lot of uh, such diseases where we did not have any uh, medical therapy or any medicine or any vaccine. In these kind of cases, gene therapy has become a good option in some of the cases, and there is active research going on, a lot of research going on also, to try and solve some of the diseases where we do not have any particular medicine or vaccine. So, although we have started using it and we have been looking towards solving some of the issues with regards to gene therapy, but we do see that there are certain limitations that we have with gene therapy. One, for example, the short-lived nature of the treatment. Now, short-lived nature of the treatment is mainly about the fact that many a times the gene therapy that is introduced to the body may not be effective for very long, and the reason for that basically is how exactly the body itself works, how the cells in the body work. And that is where you'll see that if you consider our body, every second the cells in our body are getting replaced. They're getting replaced with new cells. So a cell in its lifetime will start growing, it will develop, and then it will die. And as it dies, it is replaced with a new cell. And it is an ongoing process in our body. And so much so that if you just consider how fast it happens, by the time I would complete one sentence, almost 10 lakh cells in the body would get replaced. So that's the kind of pace that we are talking about, that within a few seconds, there are more than a million such cells which are getting replaced. Now, because of that, because of this pace of replacement, many a times the gene therapy that has been introduced to the body, that also gets lost. The body forgets about whatever new introductions had happened and goes back to its old ways of working. And that's why somebody who's undergoing gene therapy might have to go through gene therapy multiple number of times. So that's why there is one concern that we have with gene therapy, that there's a short lived nature of gene therapy. Similarly, the immune response. Many times, the immune response itself is responsible uh, for gene therapy not being very effective. Because for our immune system, the things are very simple. Anything that does not belong to the body, anything that is foreign, is an enemy. So, anything that is foreign is an enemy, and this is how they see the things. Now, in this scenario, even if, let's say, genes which are going to be helpful for us are introduced in the body, the immune system does not care. The immune system does not care, and it will simply just kill or try to destroy any genes as such many a times. So in these kind of scenarios, what then happens is that gene therapy may not remain uh, just effective for very long, or it may be it may be destroyed by the immune system itself. So that's why immune system or immune response itself becomes a challenge many a times. Then there are some disorders which are multi-gene disorders, where not only one gene but multiple genes are getting affected at the same time, and hence we are not able to use gene therapy. So whether it is about heart diseases, high blood pressure, Alzheimer's, arthritis, diabetes, in all these cases there is not one gene but multiple genes getting affected. And because of that, carrying out gene therapy against these diseases becomes very, very complex. Because if it was one gene, it is still doable. Even then, we see a lot of complexity and a lot of instability. But with multiple genes, the things become extremely difficult. And that is where uh, we see that gene therapy might not work very well. Then, lastly, another problem also is that right now it is expensive. Right now it is expensive, and it being expensive also is a big challenge. And for a lot of people, these kind of therapies are not a viable option at all, because they cannot afford these treatments. So that's why there are all these concerns that we have. And overall, the technique also remains risky right now, and we do not see I mean, we have seen that some cases it has been successful, and there are some cases where it hasn't been successful. So that's why we have to understand all these things, and we have to take gene therapy accordingly. Yes, there are a lot of improvements that have happened over the decade, and we do see that there are ways and means in which we have been able to stabilize gene therapy. But there is still, I would say, a long way to go before we can use it as a proper medical therapy. Then there is this concept which has been on and off in the news, and the concept is of three parents baby. Now, when we say three parents baby, what we see here in this particular scenario is that this is a procedure that can be used when the mitochondria of the mother are defective. Now, what happens with mitochondria is that we discussed about mitochondria, powerhouse of the cell, produce energy, provide energy to the cell. Now, if the mitochondria, when it comes to mitochondria, what we see is that mitochondria are always transferred from the mother to the child. All right, they're always transferred from the mother to the child and never from father to the child. And that's why if the mitochondria of mother are defective, then in that case, the same defective mitochondria might also get transferred to the child. So this is a situation that we want to avoid. And to avoid this situation, we have this procedure called as the three parents baby or 3% in vitro fertilization, pro-nuclear transfer. There are different names that we use for these, or mitochondrial replacement therapy. So there are all these names that we use. So overall, when we talk about this concept, it's a concept which tries to solve a problem. And the problem is, if the mother's mitochondria are defective, if they are diseased, we do not want the same mitochondria to be transferred to the child. And this is where it will play a very, very important role. This procedure will play a very important role. So that's why in this particular scenario, this is what we exactly try, that we have mother's mitochondria which are unhealthy, so we would want to replace these mitochondria with healthy mitochondria, and we can get this healthy mitochondria from a donor. So what we do in this process is that we take the donor egg cell, and from the donor egg cell the nucleus is removed, and this nucleus will be replaced with mother's nucleus. And why do we do that? We discussed something similar in case of cloning as well, that we want all the specific characters to come from this parent, the original mother. Correct? So that's why we are doing all this, to ensure that the mitochondria of mother are uh, getting replaced. So that's why we'll take the nucleus out of mother's egg cell, and then it will be placed inside the donor egg cell, and hence uh, the nucleus has been taken away and you now have healthy mitochondria. Now also understand that why are we doing this indirect process? Why did not we just replace the mitochondria in the mother's cell itself? The reason for that is that in any given cell there is only one nucleus, but when it comes to mitochondria, the number of mitochondria can be than are many. So that's why it's easy to replace one nucleus. Also, it's an established procedure already with cloning. So that's why easier to remove and replace nucleus than to try and replace all the mitochondria in that cell. So that's why we take the donor cell, we take out the nucleus from there, and nucleus of the original mother is inserted, and then the fertilization will happen, and embryonic development will start happening inside either the original mother or a surrogate mother. So in this particular scenario, we preserve the characters of original mother and father while taking care of the disease. So this is what the concept is. So so in case of this particular technique, this is basically, this technique is used when mother's mitochondria are diseased or defective. And one more point becomes important, that mitochondria are always transferred from the mother to the child and not from the father. right? So that's why, if even if father's mitochondria are defective, it doesn't really matter because that will not be transferred to the child. But if mother's mitochondria are diseased, then it matters. So that is, this question was asked in 2017 in the context of recent advancements in human reproductive technology. Pro-nuclear transfer is, now pro-nuclear transfer basically is the same procedure where we are talking about replacement of mitochondria. So that's why the correct answer here will be, I'm not launching the poll because it's a very direct thing, prevention of mitochondrial disease in offspring. So this is what we do in this particular procedure. Now, another very, very important concept is about GM mustard. GM mustard, something that has been in the news constantly. Hmm. Sorry, I forgot to tell you about this news. This was a news last year that for the first time, a baby has been born with biological parents' DNA and mitochondria from a donor. So, first case of mitochondrial replacement or three parents baby concept has been now observed, and that's why this was in the news last year as well. Now, coming to GM mustard, and again something that has been constantly in the news, GM mustard. Now, when it comes to GM mustard, this has been in the news, you can say, more or less since since 2018. Now in 2018, why it was in the news? Mainly because GM mustard seeds were about to be released for the Indian farmers, were approved by the Genetic Engineering Appraisal Committee, and the approval was also given by the government of India. And after all these approvals, the Supreme Court had put a stay on the use of or all the release of GM mustard. So just try to understand that what happened, what was this entire case about. And again, it has been in the news in the past 5 6 months. So that's why exactly it has come back in the news, and what are the problems that we have seen with GM mustard. So all the controversy that we have seen around GM mustard. So GM mustard, or what we call it as, is Dhara mustard hybrid 11, or also called as DMH 11. So this DMH11 is a genetically modified mustard hybrid variety. Now, in this particular scenario, this has been under development since almost 2004 2005, and has been developed at Center for Genetic Manipulation of Crop Plants, which is located in Delhi University. It is located in Delhi University. So this is where the development has been happening since 2004, almost since 2004. And then uh, a lot of field trials have also happened. I think between 2011 to 2017, a lot of field trials have been done. And this is where we have found that it can give up to 25 to 30% better yield than the best variants of mustard that are grown in India. So after all these things found, uh, one thing that we wanted to solve for is the import of mustard. Now in India, you know that mustard consumption in India is very huge, whether as mustard oil or mustard seeds. But we know that every part of the country, in one way or the other, consumes a lot of mustard. Now, uh, to cater to the needs of mustard in India, what you'll find is that a lot of mustard is imported. A lot of mustard is imported in India. All right. So it's not that we are growing 100% of the mustard. Majority of mustard, large amount of mustard is actually imported. More than 50%. I think approximately 60% of mustard in India are imported. So we wanted to solve this problem, that there the demand for mustard in India is very high. We have to import a lot, which also increases the price of mustard and also has a dent on forex reserves, because you have to do international trade and pay in dollars to just get that mustard. So this is where the thought process was, that let's introduce this GM mustard, which has a better yield altogether, and maybe will be able to help with the productivity of mustard and solve the problem of low availability of mustard or in India. So this is, with all this thought process, in 2018 it was cleared by the Genetic Engineering Appraisal Committee, and then it was also cleared by the government of India. and was about to be released. And this is when Supreme Court intervened and did not let GM mustard be available for the Indian farmers. And then in this particular scenario, there are some problems, or there are some issues that were brought into notice. First problem that Supreme Court had talked about was about if it can have a negative impact on human health. That because you are doing genetic modification, can this genetic modification lead to some kind of complication in the future? So can it lead to some kind of health issues in the future in humans? And because we have done a short term study, we need to do a longer study to understand if it will have any negative impact on the human body. It should not be that 5 years later or 10 years later we find that there are certain issues that are developing, and the development of these issues is because of GM mustard. So that shouldn't happen 10 years later. So that's where it was said that GM mustard uh, should be GM mustard should be uh, analyzed better, studied better, and then only we should release it. That was the first concern. That concerns that were raised Concerns that were raised first was with regards to possible health impact. Possible health impact. And second concern was with regards to the loss of biodiversity. Now, in what sense are we talking about loss of biodiversity? In India, there are, as documented, there are 66 variants of mustard that are grown in India. 66 different variants of mustard. Now, once GM mustard is available, obviously a lot of these mustard will not be cultivated anymore, and over a period of time a lot of these variants of mustard might go extinct because they're not being grown, the seeds are not available, and over a period of time you might lose on that mustard. So then the species diversity of mustard will start to decrease, and that's always bad for any species. For the survival of any species, diversity or biodiversity is very very important. Whether genetic diversity or species diversity, it is very very important for the survival of any species. And that's why for survival of mustard also, in that sense, it's important to have a lot of different variants of mustard. So that was another concern that was raised by a lot of environmentalists in case of GM mustard. So with regards to all these concerns, it was uh, there was a stay that was put by uh, the Supreme Court, and then a committee was set up so that this committee should study and analyze GM mustard in detail again and should come back with its report. So now finally in 2022, the committee came back with the report and said that no, we did not really find any problem with GM mustard with all these different parameters that we tested on, and hence we are okay with the release of GM mustard. And this is when it again went back to the Genetic Engineering Appraisal Committee, and it was cleared by the Genetic Engineering Appraisal Committee. And then it went to the Government of India, where Government of India has asked for Supreme Court's permission to go ahead. Now, this is where right now we are stuck, that uh, field trials are likely to be delayed because the Supreme Court still hasn't given any uh, re- hasn't said anything on GM mustard. The dates still haven't been given, and the deadline for growing mustard maximum can be December first week. So since it did not happen by December first week, it simply means that now the next sowing season, next winter, is when the field trials of DMH11 might start. So before that, Supreme Court needs to give a go ahead so that they can move and release it for the trials to begin. So that's why we have to see whether that happens and what happens. There is this one article. I mean, if you can go and read this article, it's a wonderful article to read with regards to GM mustard and everything related to why GM mustard, and it was there in the Hindu on September 16th. So if you can just go and just read it once, it's a wonderful article to read on GM mustard, and every aspect of GM mustard has been covered in this article very well. Please read this question. This was asked in your paper. This was asked in 2019 prelims. I'm launching the poll for you. It's an easy question because of one statement. There is one statement that makes this question easy. I hope you see that. Wonderful. 71% of you have given the correct answer. Let me discuss this with you. Genetically modified mustard or GM mustard developed in India. Consider the following statements. First is GM mustard has the genes of a soil bacterium that give the plant the property of pest resistance to a wide variety of pests. Now, we did not discuss about anything related to pests, but what we have discussed is that with this hybrid variant, we are able to improve upon the yield. Now, but this is not related to pests. This was mainly about BT cotton. In case of BT cotton, this is what it was, and also it's not about a wide variety of pest. There also, it was only one pest against which you were getting that resistance. Then GM mustard has the genes that allow the plants cross pollination and hybridization. Again, we have not discussed anything about this, but look at step number 3. GM mustard has been developed jointly by IARI and Punjab Agricultural University. This is where it is wrong. What did we discuss? That it has been developed at Center for Genetic Manipulation of Crop Plants, which is in Delhi University. So that's why this is a wrong statement, and this is one statement from where you get your answer. This is where you get your answer from, and that's why the correct statement is only two. GM mustard has the genes that allow the plants cross pollination and hybridization. It is a mustard hybrid variety, and it is something that is capable of cross pollination. And this was also raised in the Supreme Court that if, let's say, because it is possible that cross pollination can happen from this GM mustard, it might also transfer its genes to some of the other crops also growing in the nearby areas. So that's why it might become a problem altogether for all these crops as well. So that's why we need to be aware of all these, and so that we take a decision accordingly. Now, coming to the last topic of discussion in this chapter, and this is dark biotechnology. Now, in case of dark biotechnology, something that we discussed yesterday, that dark biotechnology is about misuse of biotechnology, right? It is about misuse of biotechnology. So, whether it is the act of bioterrorism or biowarfare, biocrimes, anti-crop warfare, in each and everything, and there are other ways in which we can understand these limitations. So all these limitations, the misuse, the negative aspects of biotechnology, all this will be called as dark biotechnology. Now, in this particular scenario, when it comes to dark biotechnology, what we see is that the act of bioterrorism, for example, can be counted as dark biotechnology, where there can be a deliberate use of a biological agent like a microorganism to do something against humanity, maybe cause death of humanity or cause disease in humanity, or if not, create such pests that can destroy the crops. Even doing that is possible. And if you consider a country like India, where we are so significantly dependent on agriculture, if, let's say, tomorrow there is an act of bioterrorism where some kind of crop is getting harmed, right? Some kind of crop is getting harmed, and that's why the productivity drastically can dip. And if that happens, then in that case it has a huge economic problem for India, right? So that's why in all these cases, these all will be counted as acts of terrorism. All these will be counted as acts of terrorism, and we need to ensure that these things can be countered. Similarly, regenerating extinct pathogens. Now, we have seen very recently, last year itself, when a virus was revived in the Arctic. An Arctic virus was revived by the scientists, and this is a virus that has been sleeping for 48,500 years, and this was an a virus which can be potentially deadly, but it was revived by the scientists. I don't know why, but they did. But these are the scenarios where the things can take a wrong turn, because we know that there is something like this which is possible, that we can actually revive extinct pathogens, and there are ways to do this, and this is where it can become a very dangerous scenario going forward. So that's why uh, these kind of things we have to be very uh, aware that this all can lead to a this can all take a very negative turn, and forget about these viruses being found and being revived. But at the same time, there are diseases which have gone extinct, or let's say the diseases which have no effect on human population anymore, because wide vaccinations have happened against them, whether you consider something like a plague or something like smallpox. So wide vaccinations have been done and eradication of these diseases have been done. But what about the future? Today, vaccinations are not done against smallpox, right? So today, if a pox virus starts affecting human population, then in that case, the immune system will not be able to respond properly, because now the vaccines are not being given. Correct, the vaccines are not being used anymore. So that's why the present population will not have the immunity against smallpox. And what if smallpox virus is unleashed on population tomorrow? Then in that case, it can take a negative turn. Yes, of course, we have the vaccine now because we have developed vaccines once, so it will be uh, put to use very quickly, but we at least know that there is uh, no answer that the human immune system has in these kind of cases. So that's why we need to be very aware that something like this also is a possibility, and it can lead to dwindling of it can lead to loss of lives and a lot of issues worldwide. Then the effects that it can have on biodiversity. We discussed about GM mustard, the example of GM mustard, and how it can lead to loss of biodiversity. But it's not only about the loss of biodiversity in the sense that what we are seeing in case of GM mustard, but overall the use of biotechnology is being done largely on what? Largely on only certain plants and animals, because we find that these are the plants and animals which are so called useful for us. So the ones which are not useful, which are, you can say, 99.9% of all the species that live on the earth, we are not doing anything for them. We are not doing anything to save them or to preserve them. We are not doing anything. Correct? The conservation activities are not really about each and every one of these species. It's about only a few species. So then in that case, we are protecting some species and we are letting go of a lot of different species as well. And that's why it can have a negative impact on biodiversity, because there are only few plants and animal species where all the research and everything related to biodiversity, biotechnology is being done. So that's why it can have a negative effect on the remainder species. Similarly, there is this concept called as terminator genes. Now, in case of terminator genes, these are genes which are added to the seeds in a manner that the seeds can germinate only once, meaning that in the next sowing season, suppose that as a farmer, you go to the market and you bought some seeds. Now, let's say tomato seeds. and now you grow tomato from these seeds, and you see very good quality and quantity of tomato. So then you think that, okay, because the produce was very good, you would want to preserve the seeds from this growth, the tomato growth that has happened this season. You would take the seeds from these tomato and preserve it for the next season because of the quality and quantity that you have observed. Now next sowing season, when you go back and sow these seeds, you don't do you see that the seeds do not germinate. The seeds do not germinate at all. Why? Because in the previous seeds that you had bought from the market, there were terminator genes which were placed inside the tomato seeds. And whenever you have terminator genes, when in whatever species, in whatever seeds you have terminator genes, it would mean that the seeds would germinate only once, and in the next generation the seeds that you have gotten from these fruits will never germinate. So that's why this becomes a very dangerous trend, because it is something, uh, a terminator gene in a genetically modified crop plant stops the plant from releasing fertile seeds. So that's why the plants will not produce fertile seeds, and hence these seeds cannot be used in the next season. And then the farmer will have to go back and purchase the seeds again from the market. By this time, obviously, the market prices of these seeds would have increased, and the companies would also want to make farmers dependent on them. So that's why there are all these unethical practices which where it comes to terminator genes. And it's not only about this particular crop being affected, but it goes beyond that. What if this plant that has the terminator gene also cross pollinates with another variety of crop being grown in the same area? So then the terminator genes can be passed on from this crop to that another crop as well. So then if that happens, the other crops also will have a negative impact, will see a negative impact. And even if even in their case also, we might see that in the next season they also show the same problem, that they also do not germinate, their seeds also do not germinate. So that's why there are all these issues with regards to terminator genes, and that's why we need to be aware of what all problems it can create. So that's why these are things regarding the negative aspects of biotechnology. Now, at the end of the chapter, looking at some of the questions that have come in your previous year papers in your mains examination, as I told you earlier yesterday, that the questions are not that difficult when it comes to mains examination in all these questions. So if you look at the first question right here, what are the research and developmental achievements in applied biotechnology? How will these achievements help to uplift the poorer sections of society? Now, first part is talking about applied biotechnology. Applied biotechnology is nothing but everything that we discuss in this chapter, that when you are using biotechnology for one thing or the other, in one sector or the other, that is what applied biotechnology is. So what are the research and developmental achievements in applied biotechnology? Now, again, there are so many things that we have discussed in this chapter which can be used here, whether it's about BT cotton or biofortification, or the medical applications where virotherapy has been used, biopharmaceuticals we discussed, gene therapy we discussed, stem cell therapy we discussed, DNA, the applications of DNA fingerprinting. So there are so many things, all these can be counted as achievements. So basically, you have to summarize this entire chapter that we discussed. And how will these achievements help to uplift the poorer sections of the society? Now, think in two ways. One, from agriculture and food security perspective, that biotechnology helps you produce more, get better productivity. So it helps the farmer grow better, helps the farmer in getting better value. At the same time, also can be used in regulating the prices of food or decreasing the prices of food, keeping the prices of food in check. So that's why it will have a positive impact on the society from that perspective. Similarly, at the same time, we see that in case of medicines also, cheaper medicines are now being made available because of biotechnology. So biotechnology has helped there as well in drug management. Then at the same time, there are a lot of opportunities opening up, and it is giving better opportunities to a lot of people who are associated with all these areas. So that's why there also we see that it can help in uplifting of uh, uplifting of the poorer sections of the society. So the number of points here is huge. I mean, 250 words may not be even enough, right? 250 words may not be enough, because there are so many things that you have to write. So that's why you'll have to be very picky in making uh, or writing these answers, and many a times these kind of answers can become very tricky, because there are so many things that can come in your mind while writing this answer. So you'll have to be very crisp. Try to not make stories out of each and every point. Try to be as crisp as possible, and include as many points as possible while writing this answer. But this becomes a tricky answer, not because of difficulty in the question, but because there are so many points that can be potentially used in writing answer to a question like this. Then the second question: how can biotechnology help to improve the living standards of farmers? Now, improving the living standards of farmers is also about nothing but the agricultural applications. And we discussed about so many agricultural applications. Just that you cannot directly just list down all the agricultural applications. You have to uh, change your language. You have to change your language in a manner that you are, with whatever points you are using, you are associating each and every point with farmer. That how does the farmer benefit from a point like this? So this is what you'll have to do in writing this answer, that although you are going to use the same points that we discussed in the chapter, but your language has to be different while writing that answer. Then why is there so much activity in the field of biotechnology in our country? How has this activity benefited the field of biopharma? Now, why so much activity? Because we see so many advantages. We know the potential, we see the potential, and that is why we are saying that there are so many things and there are so many activities happening in the field of biotechnology in the country. So again, you can write in the terms of medical applications and agricultural applications largely in this particular scenario. And then how has this activity benefited the field of biopharma? Now, in biopharma, how are we using biotechnology? So again, the same points: pharmacogenomics, pharmacokinetics, how in biopharmaceutics it helps in making better drugs. Even you can write about virotherapy. So there are all these things that you can include in the second part of this particular answer. So again, a simple question directly based on what we already have discussed. Then this question: stem cell therapy is gaining popularity in India to treat a wide variety of medical conditions, including leukemia, thalassemia, damaged cornea, and severe burns. Describe briefly what stem cell therapy is and what advantages it has over other treatments. Now, understand something about this question. If you look at this first part of the question is not really a question, but a statement praising stem cell therapy. So, in a manner, you can say that this is a question which has a very positive tone. Correct? It has a very positive tone because it is talking about everything that stem cell therapy can do. And then describe briefly what stem cell therapy is and what advantages it has over other treatments. Now, this is where the question starts. Now, when it comes to a question like this, because of the positive tone that we have, it doesn't talk about what are the problems with stem cell therapy, what are the limitations of stem cell therapy. It does not say anything, or does not ask anything negative about it. So while writing an answer to a question like this, you should not go negative. You also have to try and restrict yourselves to as many positive points as you can write, and talk about the present and the future of stem cell therapy. You can talk about limitations in a couple of sentences, but give a strong way forward after that. That you can say that yes, there are so many advancements that have happened in this field, but still there are areas where we need to improve. You can give certain examples, and then you can say, uh, start talking about the future and how the future of stem cell therapy holds up. So that's why you have to be as positive as possible in an answer like this, because this question itself is entirely positive, and it is not talking about anything about the negative aspects of of stem cell therapy. All right. So this is about this chapter. Before going to the next chapter, if you have any doubts, if you want to ask anything. What are zombies? Zombie is a fictional concept. At least for now it's a fictional concept, where we are saying that the for humans it's a fictional concept, although in other organisms we can say use the word zombie, where we are saying that the brain activity of an organism is not working very well, or it's not actively working, and the brain is not able to respond very well, and there are changes that happen because of that. How the genetic information obtained from dead bodies as the cells also die. No, so this will happen. See, DNA analysis, I'm just telling you this, that yes, the cells stop growing and stop multiplying and everything, but DNA analysis can still happen, because the cellular structures will remain for a very long time. And even from fossils, forget about dead bodies, we have been able to do DNA analysis from picking up from the fossils which are up to 20 lakh years old, up to 2 million years old. So forget about only a dead body which has been, let's say, dead for a while. So so that's why it's not that see, cells will stop growing. That's a thing. Cells will stop growing, they will stop multiplying, but it's not that you would completely disintegrate and everything will completely go. The remains of the DNA will remain for a very long time, and that's why we have been able to find DNA samples. We have been able to do DNA analysis of samples that are almost 1 to 2 million years old. What is mRNA? mRNA basically stands for messenger RNA, and messenger RNA is a kind of RNA which carries the message or instructions for making proteins in the body, or making proteins in the cell. So that's why messenger, because it carries the message or the instruction for making those proteins. Work of stem cells, we discussed that entirely yesterday, in stem cell therapy, in tissue engineering, in disease modeling, in drug testing, in then we can use it in regenerating body parts or organs. So there are all these things. Plus we also use them in autoimmune diseases, against neurodegenerative diseases. So multiple ways in which we can use them. Please explain how can genetic information be misused for commercial purposes. So basically, just understand what can happen is that they can take the genetic information, they analyze the genes, and if there are certain genes which are, let's say, related to a disease, even remotely related to a future onset of the disease, they will amplify that information in a manner and sell that information in a manner that it looks like it's going to be a big problem, and they sell that problem to you, and in a manner solutions for those issues. So this is what they can do, and medical packages can be sold in that manner, that they will first tell you that okay, this can go wrong and that can go wrong, and on the basis of that they'll tell you that you take this medicine, that medicine, go through this therapy and that therapy. Can you again explain how applied biotechnology helps to uplift poor in the society? So mainly just think of two aspects. One of agriculture. Now in agriculture, because of biotechnology, we have been able to get better productivity. And not only these specific examples that we saw in the chapter, if you just go and look at the number of such examples in real life, you'll find that there are a lot of ways in which we have used biotechnology. It's not only about genetic modification, but even the hybrid varieties that we have produced, they have been able to give us better produce altogether. So that's why first in agriculture we are able to cater to the food requirements from with the help of biotechnology, and that has helped in keeping the food prices in check. Then for the farmers it has been beneficial, because it has given the farmers better productivity and hence better money. So overall farm incomes have increased, and that's why helping the poor sections of the society. Then, apart from that, in case of medical science, we have seen that a lot of generic drugs are now available, and a lot of generic drugs have been available due to uh, the advancements of biotechnology. Similarly, vaccines. In case of vaccines also, we have seen something like this, that in case of vaccines also, biotechnology has ensured that faster vaccines can be made available, cheaper vaccines can be made available, and hence it can overall take care of the health of the Indian society as well. So that's why there are all these things that you can say in this particular scenario. How can we know that there is a defect in mitochondria? See, in case of mitochondrial defects, there will be, I mean, it can, the basic blood scans and blood samples that are collected at the time of pregnancy, that can many a times give an idea, or even before that. In general, the vulnerabilities, they show up in one way or the other as well. So that's why vulnerabilities will already be known, and that's why these mitochondrial diseases can be detected very early, and then after that we can take care of it in these kind of procedures. Terminator genes presently, which we see in some varieties of papaya without seeds in it. So it's not really, I mean, in this particular scenario, I wouldn't say that it's all about terminator genes. There are hybrid varieties also where we see that these hybrid varieties, they become infertile. So in case of papaya also, that is where the cases are, that uh, that papaya varieties, which are the hybrid varieties of papaya, they actually are infertile, and that is why it's not actually because of terminator genes, but because of the hybrid varieties, the way hybrid varieties have developed itself. So I wouldn't say that it's a case of terminator genes. What are biogrids and biosensors? See, we did not really discuss anything about biogrids, but in case of biogrids, what we say is that this is where we are talking about a database, a biological database, from where we can get the genes and the proteins, and it can help in modeling of the genetics. Is it true that we are importing oil derived from GM crops? And sir, what are biosignatures and how are they related to biomarkers? See, in case of importing oil derived from GM crops, there have been uh, certain researches that were done, certain studies that were conducted. The studies were conducted on soya bean largely and soya bean oil, etc., and where we found that it, in some of the soybean oil that was being sold in India, in the study was conducted in the Delhi NCR region, and the study that was done there, they found that the soya bean that is being imported from Southeast Asia, from US, from other parts of the world, they actually were GM soya bean. So in India, on one hand, GM soya bean or GM food in total is banned, and when it comes to importing of the same food, we are seeing that imports of these foods are happening without us knowing that there were genetically modified soya bean, etc., that were being sold. And this was a study that was conducted by Center for Science and Environment in Delhi NCR, and there they found that in lot of soya bean cases, whether it is uh, direct selling of soya bean, or even if, let's say, it is about the soya bean oil, in all these cases they found it. So yes, in that case you can say that this is actually which has happened. And not only that, even on mustard also this has been seen, and in mustard also there are certain parameters where we found certain problems in the imports of mustard that we have done. Then your next question was, what are biosignatures? So biosignatures, basically you can say, is any biological characteristic, any biological characteristic that can be that can be used, and it can be used as an evidence to understand the past, the fossils, etc. So anything. So it can be any characteristics, any DNA, any molecule, anything that we find. So that can be called as biosignatures. What is IVF and why the success rate is not good? IVF stands for in vitro fertilization, related to surrogacy that we had discussed yesterday. To get success in IVF, it's not that it's completely uh, it's not successful. It has been successful. Success rates are low because we are trying to place those embryos and trying to get the fertilization done um, in an artificial manner. So many a times it becomes very difficult to carry out these processes. So that's why the success rate, because of the instability of the process, has been low. CRISPR-Cas9. CRISPR-Cas9 basically is a biotechnology tool, a genetic engineering tool, which is used in recombinant DNA technology to mark the areas where the cuts have to be made and where the insertions have to happen. So CRISPR is, you can say, the one that marks the areas where the cuts have to be made, and Cas9 is like a biological scissor or a molecular scissor that makes the cut in the required location. So it's a biological, or you can say, a genetic engineering tool. These companies just to earn profits using terminator genes. Yes, there have been cases here, and can the government ban them? Yes, government can ban them, and governments have banned them. But many times the governments do not know that it happened. The companies will not say that they had used terminator genes. So there was a big case that had happened, a lot of controversy that has happened against Monsanto, when Monsanto as a company had used terminator genes in their seeds. So a lot of controversies have actually happened already in this particular scenario. That means that there is a need to have a regular check. Yes, of course, there is a need for having regular check on the food, especially on the food imports. Many a times, the problem is that a lot of imports that happens, there is no disclosure that what they have used is something like this. So that's why this is where we see that it can a lot of times become tricky, where we see that it becomes very difficult for even the government to look into each and every data piece and track each and every one of these biological resources which are being imported into the country. But yes, there is a need for all this. There is a need for regular checks being there, and not through researches only once in a while. What is bioinformatics? So bioinformatics is when you use computers or applications of computers in a manner that we are trying to capture biological data, all right, genetic data and biological data. So that is called as bioinformatics. So when you analyze each and every genetic information, DNA information, uh, all the things about chromosomes, genomes, and everything, computations that we do mathematically, etc., all these will be called as bioinformatics. Are MNCs allowed to use terminator genes in India? See, terminator genes are not allowed to be used in any way. Just that, and even if they are going to use these terminator genes, they have to first declare that they are using terminator genes in the seeds, and then after that, it's up to the farmer whether the farmer wants to buy it or not. So largely, but we see that this is where which is this is something that is not allowed, and that's why in the past there have been a lot of controversies as well. What is tRNA? tRNA basically is transfer RNA. It is, you can say, in a manner, a link between uh, it helps in protein synthesis, and it's like a link between messenger RNA and the site of the protein synthesis, and that's why plays a very very important role. The basically the amino acid chains that we have in our body, uh, between them and the messenger RNA, they act as a bridge. They act as a link between the two, and it will help in protein synthesis altogether. I'm a student of the morning batch. Is it sufficient to join today's second class in continuation of our classes? Yes, you can do that. Mostly, uh, you can say after the break, everything that after the lunch break, everything that we are going to discuss, that will be new. if you belong to the morning batch, so you can come then. Why cervical cancer is in news? Is it dangerous? Yes, it is dangerous. It can be potentially fatal, and that's why there are vaccinations that are available against it. The HPV vaccination that should be taken. Isn't it bad that the companies are selling Terminator genes? Of course, it is bad. Of course, it is bad. Just that it is unethical. I mean, to say the least, it is unethical. Just that the governments have to know. Many a times, the governments do not know, and even if, let's say, they are not using Terminator genes in all the seeds, they might just mix some of the Terminator genes in some of the seeds, which nobody will come to even realize or know that this happened. So that's why it becomes very dangerous. COVID-19 is a bioweapon or not. We do not know for sure. We do not know for sure whether it is a biological weapon or not. That if it was made in a lab, then in that case, yes, it is a biological weapon. But if it was natural, then we cannot call it as bioweapon. CRISPR-Cas9 comes in which context? It comes in the context of recombinant DNA technology, where we discussed about restriction enzymes and the role of restriction enzymes and DNA ligase. This is exactly where CRISPR-Cas9 as a genetic engineering tool becomes very crucial. Do you teach more in depth in morning class or weekend class? Whether it is morning class, evening class, weekend class, English batch or Hindi batch, I teach exactly the same thing in each and every one of these classes. There is no change whatsoever in, I mean, there is no difference in the PPTs, there is no difference in even the language, everything will remain the same, and there's no difference that will come whether whatever batch it is. And that's why equal amount of time has been given to each and every batch. Exactly same time is allocated to each and every batch, so there is nothing to worry whether this batch or that batch. Every batch will have the same thing, whether online batch or offline batch, whether a batch in Delhi or a batch in Bangalore, each and every batch will have exactly the same thing. All right. So let's take a small break here. And on the other side, let's start with nanotechnology as our next chapter. It's 11:23. Let's be back by 11:33. Let's take a 10 minutes break and start our discussion. Welcome back, everyone. Let us start with the next chapter, and this is our chapter on nanotechnology. Now, nanotechnology, first from the perspective of your prelims and mains examination, understand this that in prelims, I would say it's more important than in mains. Although it's not that questions have not come in the mains examination, they have come in the mains examination as well. But in case of nanotechnology, it is still a chapter which is pretty oriented towards your prelims examination than your mains examination. So hence, the discussions, the way you study the current affairs will be slightly more tilted towards prelims than in mains. But we'll discuss how questions can come and what kind of questions can come, if at all, in the mains examination as well. Now, overall, when it comes to nanotechnology, understand first and foremost what do we mean by nanotechnology. That there are two words that we have here: nanotechnology. Now, the meaning of this term nano is dwarf. It's a Latin word for dwarf. And that's why for anything that is small, we tend to use the word nano. We discussed about nano satellites. So nano satellites are what? Nano satellites are the satellites which are very small. So that's why these are called as nano satellites, correct? So similarly, Tata Nano car. So Tata Nano car is another example. Why? They made a compact car, so they named it as nano. So in general, anything that is very small, nano is a word that we tend to use for it. Now, nanotechnology, although it's not only about anything that is small, but rather we are talking about the molecular levels or the molecular particles, where we are talking about the size that is 1 nanometer. Now, how much is 1 nanometer? 1 nanometer is equal to how much? Yes, 10 to the power of minus 9 meters. So now when we talk about nanotechnology, in particular, we are talking about technology of this order, technology of the order of 10 to the power of minus 9 meters. Now, why is this order or why is this scale so important? Why do we see that everything hovers around this particular scale? Why did we name an entire branch of science only on this particular scale? The reason is this is where the molecules get starting, they start to assemble. Before this you had atoms, or you had, let's say, inside the atoms, electrons, protons and neutrons. So electrons, protons and neutrons at that level, they're not different. Electrons inside a carbon will be similar to electron inside nitrogen or phosphorus or hydrogen, or wherever the electron is. So electrons, protons and neutrons, they are not different in their properties. Their configuration is different, that makes up an atom of a particular element. So that is where we see that their configuration is different, and hence this is where they are different. uh, uh, but even when it comes to the atom, we know that atom, wherever they are, they would have similar properties. But when you make a molecule out of an atom, this is where the properties are well defined in a only particular sense. Understand what I'm saying? Suppose that you have hydrogen and oxygen. You have these two elements. Now when you have these two elements and when you make a compound of out of it, you can either make water, that is H2O, or you can make H2O2, which is hydrogen peroxide. So either water or hydrogen peroxide. In both these cases, you have hydrogen and oxygen, only with one small difference, that in one case you have two atoms of hydrogen and one atom of oxygen, and in the other case you have two atoms of hydrogen and two atoms of oxygen. And this one extra oxygen changes everything, changes the entire property set that hydrogen peroxide has. Hydrogen peroxide, we can use it as an antiseptic, it is used as an industrial reagent, it can be used as an oxidizer, oxidizing agent, that is. So there are multiple ways in which we can make the use of hydrogen peroxide. And water, I need not tell you, you can drink water, you cannot drink hydrogen peroxide. Please don't try that at home. So there are a lot of differences that we have, just because there is one extra oxygen atom, right? So oxygen atom in its form, wherever it is, will be similar. But as soon as it combines in a certain form and makes a compound, this is where the properties of that compound completely changes. Hydrogen is a fuel, oxygen helps in combustion, and when they combine they make water which can extinguish that fire, correct? So properties completely change from the respective properties that they had at their atomic level. So this is where the things become very, very crucial to understand, and this exactly is the molecular level. This is the level of almost 1 nanometer. A water molecule size, for example, is 0.1 nanometers. So this 1 nanometer size are near about, especially 1 to 100 nanometers, they all will be considered as nanomaterials or nanoparticles. And forget about hydrogen and oxygen. If you, even if you consider carbon, if you consider the forms of carbon, we discussed about coal, and we understood that coal can be used as a fuel. Then I'm sure you know what a diamond is, and diamond becomes a precious stone which is very, very costly. And then you have graphite, which is also completely made of just carbon and nothing else, and you use graphite in your pencil. So one form of carbon, which is just carbon atoms, bunch of carbon atoms together, makes one of the most precious stones that is available here, and the other one makes up your pencils in your day-to-day life. So such stark differences, just because the arrangement of the carbon atoms are different. Only because of the arrangement all these differences have come in, and all these different forms where you have the same element, they are also called as allotropes. These are all called as allotropes of carbon. These are all called as allotropes of carbon. So in, so various forms, you can say in simple terms that these are various forms of carbon. So overall what we see is that when it comes to carbon, there are various different forms in which carbon can be uh, seen, and every time carbon atoms are not different, their arrangement is different, and just the difference in arrangements brings in all the difference in the properties of carbon. So this is where nanotechnology comes in. This is where nanotechnology becomes such an important technology, because we have found so many nanoparticles, we have found nanomaterials, and we have seen their properties, and we have seen what they can do. And with everything that has happened, we still say that we have just started knocking the doors of nano technology, and we have not really done anything which is going to be extremely complex use of nanotechnology. So everything that we are doing, it's just the beginning of the use of nanotechnology. So overall, nanotechnology basically comes from the Latin word for dwarf, and we are talking about 1 nanometer size and everything near about 1 nanometer size. And understand the importance that the properties that you see in any particle or anything at your level, all these properties, they first start to show up at nanoscale. And that's the reason why nanoscale is so important, because this is where the properties, they start showing up for the first time. And before this, the properties, they did not exist, they did not show up at all. So that is where we see that nanotechnology becomes important. The term nanotechnology was coined by Norio Taniguchi, a Japanese scientist, back in 1974. So it's a very new science as well, compared to space technology or various forms of energy, or even biotechnology for for that matter, where we have been able to do a lot, and a lot of research has happened over hundreds of years. In case of nanotechnology, the name itself did not exist until 1974. It's a fairly new science in that sense. So, in case of nanotechnology, what we are saying is that it's a branch of science that deals with the minute particles in the range of 1 to 100 nanometers in size. 1 to 100 nanometers. All right. So, it's not that it's hard and fast 1 to 100 nanometers. As I told you, that even if it is 0.1 nanometers, we can still consider it as nanoparticle. So overall, that's why we have seen that nanotechnology starts taking a shape in this particular scenario. And it's not only about identification of nanoparticles and these nanomaterials and what these nanomaterials can do, but it's also about the manipulation of properties that is possible. That is, there is a possibility that we can change a certain matter structurally. We can change the properties of that matter. We can bring in a positive property in a matter, and we can actually use it in various different forms. So that's why there are a lot of possibilities that we have with nanotechnology. There are a lot of things that nanotechnology can do potentially, and as I told you that we have just started using nanotechnology. We have just identified various ways of using nanotechnology. Now overall, when it comes to nanotechnology, that's why all these nano all these, you can say in a manner, they all can be considered as nano material. They all can be considered as nanomaterial. So nanomaterial because these are belonging to that range of more or less 1 to 100 nanometers. So that's why if you see, even compared to the nanoparticles, even our blood cells are much bigger. That the size of red blood cells can be 5 micrometers, which you can see in a manner is 1,000 times. It's 1,000 times bigger than the nanoparticles. So that's why nanoparticles can very easily enter inside the bloodstream or the blood cells, and they can be carried wherever we want. So that's why we say that this is a case of targeted drug delivery, that we can do targeted drug delivery with the help of these nanoparticles. So that's why there are a lot of things that can be done, and we'll discuss about all these applications as we move forward in our discussion. So overall, when we look at nanotechnology and its development, the first signs of development is in 1959. Where this lecture by an American physicist, Richard Feynman, was being delivered, and this lecture was called as "Plenty of Room at the Bottom." Plenty of room at the bottom. Now, plenty of room at the bottom. The lecture basically was about nanotechnology. In a manner, you can say about nanotechnology, because this is where Richard Feynman started saying that look, whatever we know right now, it's about everything that we can see, and we haven't really given our minds and thoughts to what we can't see, what lies beneath, and it's time that we should start looking for what lies beneath everything that we see. So this generated a lot of interest in the minds of the scientists and the students as well, because Richard Feynman was a very famous scientist, a very popular scientist as well, among the students and the scientists alike. And you can say in the last 40, 50 odd years, he has been one of the most famous scientists of the generation, and a very widely followed scientist, especially in terms of nuclear physics, in terms of quantum physics, in terms of nanotechnology. These are the areas where especially he was very widely followed, and that's why whatever he said, the scientists and the students would take it seriously. They would start doing everything that he would say, because of his popularity and what he meant for the scientific world. And he became very famous. There's a book also written, a biography on his life, called as "Surely You Are Joking, Mr. Finman." Wonderful book to read if you like reading biographies or any book like this. One of the best biographies that I would have read in my life. It's such a well written book, and the name itself is very peculiar. "Surely You Are Joking, Mr. Finman," because it has a lot of incidents from his life, which are unthinkable. That okay, that happened and this happened and that happened, and because of all these things, it becomes a very interesting read. And surprisingly, it also became one of the best sellers. And a scientist's biography becoming a bestseller is unheard of, but it did happen with the biography in his on his life. "Surely You Are Joking, Mr. Finman." So this happened. I mean, this generated a lot of interest in the minds of students and scientists, and we see that a lot of theoretically studies start happening here in nanotechnology, in molecular science, in nuclear science, in quantum technologies. We see that in all these cases now a lot of researches they start happening, a lot of theoretical studies start. In 1974, the name was coined, that is uh, nanotechnology. But even then, until now, all the studies were largely theoretical studies. Practical implementations were not yet seen. So, this is where the things change, where we see that in 1981, invention of scanning tunneling microscope happened. So, 1981 brings in scanning tunneling microscope. Now, in case of scanning tunneling microscope, why this invention was important is mainly because now you had an instrument, now you had a microscope that had a capability of looking at nanoscale, which could look at nanoscale, which could see at nanoscale. Until now, whatever we had, we did not have the capacity to look at nanoscale, but now with scanning tunneling microscope, we now generated that ability that we could now look at nanoparticles. So until now, a branch which was completely theoretical now starts becoming practical, because now we can look beneath, now we can see the molecules, we can see their structure, we can analyze their structure. So that's why scanning tunneling microscope was a big event in this entire development of nanotechnology in the world. Now understand what is scanning tunneling microscope, and there are basic principles that are behind the working of scanning tunneling microscope. So when it comes to scanning tunneling microscope, there are two basic principles that work behind the scenes. Now, one, which basically is the process, or the concept of tunneling, and then the concept of piezoelectric effect. So one is the concept of tunneling, and one is the concept of piezoelectric effect. Now these are two things that are behind the working of scanning tunneling microscope. Now, the invention of scanning tunneling microscope, it also got a Nobel Prize. And hereafter, you'll see that there will be a lot of things. I mean, almost all the topics that we are going to discuss, you'll find that every topic has a Nobel Prize in its name. Every topic has a Nobel Prize in its name. So, even scanning tunneling microscope, I think 1986, it got a Nobel Prize. So, now scanning tunneling microscope, understand what it does. If you look at this figure, what we do is that whatever sample we have, there will be a metal tip that will be incident on that sample. All right, we will press this sample, we will apply pressure on this sample with the help of this metal tip. Now as we put pressure on this sample, as we put pressure, there are electrons that from the metal enter inside this surface. All right, so suppose that you have a surface like this, suppose that you have a surface like this. So then what happens is that the electrons, they travel inside like this. They would be able to go inside the sample. In a manner, you can say that they are able to break the energy barrier and enter inside this sample. So that's why the concept is called as tunneling, that they are able to make a tunnel through this sample. Have you seen, there is a film called as Ant-Man by Marvel movies? Oh, Roshan, you have already given that example. So, this Ant-Man, if you have seen, so you might have seen. For those of you who haven't, uh, this was also a trick question. You shouldn't see films right now. You go and study after the class. So for those of you who have not seen the film, what they show there is that the main hero or the central character, he basically would be able to shrink to a size of an ant, you can say in a manner, and then enter inside something called as quantum realm. So this quantum realm is like the concept of tunneling, where you are making a tunnel, you are able to break the energy barrier, and you are able to enter inside the material. So that entering inside the material that happens in that film is nothing but the concept of tunneling. So that's why they keep saying quantum, quantum, quantum. So that quantum is nothing but the concept of quantum tunneling. So through quantum tunneling, the subatomic particles, yes, humans cannot do that. That's science fiction, that what they have seen in the film. But subatomic particles can do this. Whether electrons or photons or other such subatomic particles, they are capable, that they can undergo this concept of tunneling, they can show this concept of tunneling, and hence for them it is possible to enter inside the surface. So that's when now the electrons will be able to enter inside the surface and analyze that surface, understand that surface. This is called as tunneling. So tunneling basically is where electrons break the energy barrier of the material. They break the energy barrier of the material and enter inside. So this basically is called as tunneling. This is called as tunneling. So electrons break the energy barrier and enter inside it. So that's why it is called as tunneling, that they in a manner are making a tunnel through that material. So in other words, they make a tunnel through the material. So this is what happens in case of tunneling. Now the second principle is piezoelectric effect. In case of piezoelectric effect, what we basically see is that this is where when you apply pressure on a solid, you generate electricity. So generation of electricity by applying a mechanical stress or pressure on a solid is called as piezoelectric effect. Now, until now, we had seen piezoelectric effect only in solids, but last year this was also in the news that for the first time, scientists have been able to observe piezoelectric effect in liquids also. Until now, it was seen as a property of solids only, but now it has been observed in liquids as well. So just understand this concept of piezoelectric effect because it makes this concept important, because now it is in current affairs. So piezoelectric effect is when you if let's say electricity is generated by applying mechanical stress or pressure, exactly what we were doing, that with the help of this metal tip we were applying pressure on this sample. So as we applied pressure on the sample, this is what basically led to this is what led to electrons getting generated, the electrons that are entering. So electrons, generation of electrons is nothing but generation of electricity. What is electricity? Electricity is when there is a flow of charge, right? Flow of charge with the help of electrons. So that flow of charge can only happen when electrons or whenever electricity is getting generated. So here this is what's happening, that when you apply mechanical stress, if electricity is generated by applying that mechanical stress, then in that case we'll say that it is piezoelectric effect. So that is exactly what's happening there. The electrons that are getting generated, the electrons that are getting who are the electrons that are making a tunnel, electrons that are making a tunnel through the surface, these electrons were generated because of piezoelectric effect. So piezoelectric effect basically is electricity or electrons generated by applying mechanical stress or pressure. Mechanical stress or pressure. So this is basically called as piezoelectric effect. So these two, that's why these two concepts, they work in making scanning tunneling microscope possible. Then after this we have another microscope which is called as atomic force microscope. Now when it comes to atomic force microscope overcomes a problem that we face with scanning tunneling microscope. And in case of atomic force microscope, what we see is that uh, atomic force microscope overcomes a problem that we see with scanning tunneling microscope. And this problem is that scanning tunneling microscope only works on metals, only works on conductors, because you want the electrons to pass through, and electrons can only pass through if you have a conducting surface. If you have a non-conductor, an insulator, then electrons cannot pass through insulator, cannot enter insulators and all that. So that's why here those two concepts of tunneling and piezoelectric effect will not work if you have a non-conducting material. So for non-conducting material, we see that for non-conducting material, we see that this is where we see that atomic force microscope, it becomes useful. Now, in case of atomic force microscope also, and as I told you that every topic that we'll discuss has a Nobel Prize for it. So atomic force microscope also got a Nobel Prize. It also got, I mean, the Nobel Prize, I think it was 1986 Nobel Prize, which was shared between the scientists who invented scanning tunneling microscope and who invented atomic force microscope. So they got half and half for getting these microscopes to work on nanomaterial. So atomic force microscope overcomes a problem that scanning tunneling microscope has, that it can be used only on conducting surfaces, whereas in case of atomic force microscope, it can be used not only in it can be used for non-conducting materials as well. So that's why they also become pretty much useful. So in this particular scenario, what we see is that there is a tiny probe, the tip and the cantilever, which will scan the surface. It will scan the surface and it will try to analyze the surface by scanning through this surface. So that is where we see that this atomic force microscope will work. So that's why it will works on non-conducting material where scanning tunneling microscope has a limitation. So this is where we see that atomic force microscope becomes useful. So now with by having these microscopes, now we see that there are wide variety of wide variety of analysis and studies could be done with the help of these microscopes. And then a discovery happened, and this discovery was a discovery of fullerene in 1985, and then in 1995 this also got a Nobel Prize. So now fullerenes, when the discovery of fullerenes happened, the fullerene discovery was very very important because it was seen as the third form of carbon along with graphite and diamond. So you can say another allotrope in a manner, that it is something that we call as another allotrope, another form. So now in this particular scenario, what we observe is that these fullerenes, once these fullerenes were discovered, it was not only about the fact that a new particle was discovered or a new material was discovered, but at the same time, the properties that we saw in fullerenes were exceptional. We saw that they gave extreme durability. They saw very good uh, conduction of heat and electricity. uh, their strength was very good. They could act as radical scavengers. We'll discuss about what are radical scavengers later. But we saw all these properties, and all these properties became extremely important because we understood that these are the properties that will help us in making materials which we are not able to do yet. So this is the structure. This kind of fullerene, this is also called as Buckminster fullerene. This is called as Buckminster fullerene, where you have 60 carbon atoms joined together like a sphere, right? Like a football, where all these balls that you see, you can say that these are the carbon atoms which are joined together, as you can see, in hexagonal manner. Largely they are joined in, almost all of them are joined in hexagonal manner, some pentagonal as well, but largely hexagonal. So you have hexagonal arrangement of carbon atoms which has in totality arranged like a sphere. And that's why in this particular scenario we see that this fullerene discovery became a very important discovery because we had never seen anything like it before. And also the properties that we saw with fullerenes, we understood that there are a lot of ways in which fullerenes can potentially be used in the future. Now after this, the second was synthesis of carbon nanotubes. Now these carbon nanotubes also become extremely important because here if you look at the structure, you still have the hexagonal structures, right? still have the hexagonal structures, but these hexagonal structures are now arranged in a tube like form or in a cylindrical form. So this is where we see that we have a new kind of an arrangement, and this is also nothing but carbon. Here also you have nothing but only carbon. So here also we see that it became a very important synthesis, because we see that this is where carbon nanotubes actually showed a lot of potential. And the way it is, the structure that it is, we saw immense potential with it, because of all the properties that we observed with carbon nanotubes: high thermal and electrical conductivity, very good optical properties, flexibility, increased stiffness, high tensile strength, even up to 100 times stronger than steel in the sense of per unit weight, and at the same time being very lightweight, and hence you can also manipulate it while it remains strong. So in this scenario we see that there are so many properties of carbon nanotubes which can become potentially useful in a lot of different ways, and that is where we saw that carbon nanotubes could become very crucial. And that's why when we look at the applications of carbon nanotubes, there are a lot of different sectors where they can be useful. So first, for example, electronics. In electronics we know that they can be used to create highly conductive material, and at the same time they can also be used in making memory cards, or even, I mean, computer storage or computer chips, or even flexible displays. Today you see that there are phones which have flexible displays, so in making those flexible displays also carbon nanotubes can become useful. So overall in electronic devices we find that they can become very very useful because of their conductivity, and that will help in creating all these different types of electronics. Similarly, in the sector of energy also we see that this becomes very very crucial, because they, first of all, they have very good conductivity, and at the same time they also have large surface area. And because of this they can be useful in making a lot of different energy storage devices. So for example, lithium ion batteries, fuel cells, solar cells, they can be used in making all these because they have high surface area and they have very good conductivity. So because of that we see that in all these energy storage devices they can become useful. Then they can also be used in biomedical applications, where they can be used. If you remember, we discussed that these nanoparticles can be used in drug delivery, where you can insert uh, carbon nanotubes and make them the carriers of drugs and genes. I mean, even in genetic engineering, carbon nanotubes can become important, because you can carry these small particles, or you can carry the particles of medicine, or carry the particles of genes, and uh, you can actually use them in drug delivery. So that's why then these nanoparticles can be inserted inside the bloodstream or inserted inside the blood cells, and the blood cell will carry them to the required location. So then in that case carbon nanotubes can be useful there. Even this has a Nobel Prize. 2016 Nobel Prize was given for making molecular machines, and these are nothing but molecular machines. So 2016 Nobel Prize in chemistry is what went to making molecular machines. So that also we see that is where in biomedical devices they can be useful. Then making a lot of optical instruments, etc., in imaging applications they can be useful. Even in case of heart failures, cardio failures also, they we see that there also they can be made use of. So carbon nanotubes can become very very useful in all these different scenarios. Then in making composite material, where we are trying to enhance the strength of a material, there also we can make the use of carbon nanotubes. Whether it is a metal device, whether it is polymers or ceramics, in all these cases carbon nanotubes can be mixed, and by mixing carbon nanotubes we can enhance the strength of the material and also make lightweight materials at the same time. That you have very lightweight material which are very strong, because you are mixing carbon nanotubes in them. So that's why they can be added to polymers, metals, or ceramics to create strong, lightweight, and heat resistant materials. So that's why composite materials, we can make composite materials out of them. Then they also have environmental applications, because this is where they can be used to create filter membranes, because again they have large surface area. So that's why they can be actually used uh, in making filtration devices, and they can help in removal of contaminants from water. And then at the same time they can also be used in making sensors, so that you can detect pollution, or you can detect the pollutants. So there are all these things that we can do in terms of the use of the carbon nanotubes. And then they also have aerospace applications. So aerospace applications, because we want to make strong aircrafts and strong space vehicles, at the same time very lightweight vehicles, so that you decrease the fuel consumption. So for decreasing that fuel consumption also, we can make the use of carbon nanotubes. So there are a lot of ways in which we find that we can make the use of carbon nanotubes. At the same time, in the future, we find that there are a lot of ways where we'll still be able to find ways in which carbon nanotubes can be used. There still are ways in which we are finding out the ways in which they can be used. There are a lot of studies, a lot of research happening in various different sectors and fields, where we are trying to understand how exactly can we make the use of carbon nanotubes. There is this one question that was asked in your prelims examination in 2020. Please read this question. I hope that you've read the question. I'm not launching the poll for this question, and I'll tell you why in a few moments. What do you think the answer should be? Can you tell me in the chat? Hmm. So a lot of C's and D's, and some one or two cases of A's also. All right. So I think that couple of you have attended my class before. Anyway, let me discuss this question. With reference to carbon nanotubes, consider the following statements. First, they can be used as carriers of drugs and antigens in the human body. We already discussed this, that this is doable. Already we have a Nobel Prize that has been given to it. So one, definitely correct. Second statement is that they can be made into artificial blood capillaries for an injured part of human body. Now, whether or not it can be done, you have to understand how the question has been framed. They can be made into, it says can be. Now, until now we haven't done it. Until now, if you ask me whether we have been able to make artificial blood capillaries out of carbon nanotubes, the answer is no. Until now we haven't. But can we do it? Potentially yes. There is something called as tissue engineering, and we'll discuss this later also. We discussed about this in biotechnology as well. So in tissue engineering, we also have been trying to use carbon nanotubes, and with the help of carbon nanotubes we have been trying to make artificial tissues or implants, etc. And in organ implants we have been trying to use it. So making artificial blood capillaries in the future may be possible. So that's why when the statement says can be should be marked correct, because if you mark it as wrong, you are saying that they cannot be, means that we'll never be able to do that, or we can never make it into artificial blood capillaries, which will be, you can say, extreme to say that we cannot do it. So That's why I would say that we should mark it as correct. Third statement says they can be used in biochemical sensors. They can be used in biochemical sensors. We have used it in biochemical sensors. NASA has various biochemical sensors that have been based on carbon nanotubes. So that's why 3 definitely is correct. The fourth statement says carbon nanotubes are biodegradable. Now, this is where I would say that carbon nanotubes are not biodegradable, because carbon nanotubes are very strong, stable structures and not easy for any microorganism or any biological agent to act upon easily. So that's why uh, for destroying carbon nanotubes, we use the process of incineration, that we heat up carbon nanotubes to up to 800 degrees Celsius and beyond, and then we are able to destroy the structure of carbon nanotubes. So that's why carbon nanotubes easily, with the help of microorganisms, cannot be broken down. We have been trying to do a genetic modification in microorganisms so that we can make them to act upon carbon nanotubes, and there are various uh, studies that have been trying to do this. So that's why until now we should not say that they are biodegradable. If, let's say, this statement had said that they can be biodegradable, then I would still say that fine, we can mark it as correct, because we have developed certain microorganisms that can act against carbon nanotubes. So that's why, in my opinion, I would say that this is not a correct statement. So, in my opinion, the correct answer should be 1, 2, and 3. But unfortunately, none of the 4 statements say 1, 2, and 3. The correct answer, according to UPSC, is 1, 3, and 4. This is the official answer. This is the official answer, and that's why I'm just writing it here. This is my opinion. I don't believe 1, 3, and 4 should be correct, because 4 should not be marked as correct. In, let's say, worst case scenario, I would have marked 1, 2, 3, and 4. And this is precisely what we did. Just after the prelims examination of 2020, when on YouTube, when we were doing the live analysis of this paper, this is when we actually had given D as the option, in the event of lack of an option that we can mark as correct. So D as an option we had given, but then when the official answer came out, then to our surprise, the correct answer that UPSC had given was 1, 3, and 4. Uh, but unfortunately, I don't agree with this answer. I can't really say why they would mark 1, 3, and 4. And forget about why they would mark. Why should they even ask a question like this? This question makes no sense, because first and foremost, the statements are vague. I mean, whether they are biodegradable or not, you just go through the papers. There are so many papers that have been published in Scientific American, in Nature Journal, in Science Journal, and none of those papers you can say that okay, this is what it is. And there are so many research, so many studies happening on it. So you cannot say that they are biodegradable or not as of today. I don't know where they got this statement from, whether it was just a scientific paper that they were Googling or what happened, or even, let's say, about they can be made into artificial blood capillaries for injured part of human body. It's a pretty vague statement and very specific, because they are saying artificial blood capillaries. So that's why both at the same time. So I don't know why this question should even come. What was in the minds of the question paper setter? But they asked it. But normally these kind of questions don't come, and not only that they don't come, they shouldn't come, because it's not only about non-science background students, that it's impossible for a non-science background student to answer a question like this. Even for a science background student, it is impossible to answer this question unless they are from the field of biochemistry, they are field or from the field of nanotechnology. Then probably they'll have an idea about some idea about what should be a right answer. Even then they'll not be able to get the right answer, because they would probably also think that 1, 2, and 3 is the correct option. So unfortunately, this question did come, and in 2020 specifically, we saw a lot of vague questions, a lot of vague science questions. It looked like that whoever said the question paper was not even from, I shouldn't say that, from science background. but we don't know. Coming to the next topic, and this is about nanowires. Now, nanowires, we are talking about wires which have very small diameters and sometimes as small as just 1 nanometers. Now, in case of nano wires, we see that there are multiple ways in which nanowires can become useful, because they can be used in building tiny transistors for computers and other electronic devices. At the same time, because of their remarkable optical, electronic, and magnetic properties, they can also be used in storing computer data. So that's why in data storage, in shrinking the size of data storage, also we can make the use of we can make the use of uh, these nanowires. So that's why these nanowires become extremely important from this perspective, because they would help us in storage of all these um, storage of the computer data also, and they would be able to help us shorten or shrink the size of electronics because of all these aspects. So that's why they become very, very crucial, and they have been in the news on and off. Even 4 5 days back, there was an article in the Hindu about Elon Musk and Neuralink. So if you look at Neuralink and how it functions, there also you'll see that nanowires actually become useful in making those Neuralinks that they have started testing. So that's why nanowires, they find their use in all these. Then we have graphene. But before that, if you have any questions from everything that we have discussed in nanotechnology until now, any question if you have. Usually in science and tech prelims questions, whenever can be done is used, it is usually correct. Yes, can be kind of statements are usually correct, because uh, they leave out a lot of possibilities. So that's why they that language many times tells you that it should be a correct answer. If Neuralink is successful, will the person be allowed to appear for UPSC? Long way to go before all this can become a possibility. So we still have to see where it goes, and when they are successful, and what happens next, and when it comes to India. There's a lot before that happens. Fullerenes. So in case of fullerenes, what we are saying is that fullerenes are nothing but spherical structure of carbon atoms arranged in a spherical form, where the link between them is hexagonal in nature, and their properties have been seen to be wonderful. Their properties have been seen to be very good. I mean, all the best properties of metals and non-metals were all seen in these fullerenes, and that is where we saw that the fullerenes became a very important discovery. What is nanobubble? So nanobubbles, I think they are used in wastewater treatment. That in wastewater treatment we have these very small size nano bubbles, and almost I think around 100 meters in 100 nanometers in diameter, and they basically are used in wastewater treatment. And one of the, I mean, some of the most difficult contaminants to be which are difficult to remove, nanobubbles can be used to remove those contaminants as well. In the near future, will it be possible to shrink any object to nanoscale from its real size? I don't really see any technology out there which can do this. Can be, but in this question doesn't work. Can you tell me the reference for what you are saying? Optical wires are nanowires. No, optical wires are not nanowires. Optical wires are completely different, because they would work on a completely different principle. So that's why we cannot really say that it's. Elon is creating Neuralink now, but there is a movie called I Smart Shankar. Yeah, it's an entertaining film for those of you who like it. Uh, where Neuralink as a concept has been shown in films before, and yes, in I Smart Shankar also this was shown. Could you please briefly describe tunneling microscope? Huh. So, in case of scanning tunneling microscope, what we are saying is that we will take the sample surface, I mean, whatever material it is, and we would apply pressure with the help of a metal tip. We will apply pressure on it, and while we apply pressure, this is where we see that electrons are generated from this metal tip, and these electrons will enter inside this surface. They'll enter inside this sample surface, and in a manner we say that they are able to make a tunnel. That the electrons will make a tunnel through the surface, and they will be able to look inside and analyze what the surface looks like, and hence we'll be able to understand what the surface is. So that's why there are two concepts making it possible. One is the concept of tunneling, where the electrons are making a tunnel for themselves by making an energy barrier of that material. And then there's the concept of piezoelectric effect, where electricity is produced by applying pressure. So that's why this is what happens in scanning tunneling microscope. All right. Puri sir predicted it before. Yeah, he did. So anyway, so let's move forward. The next topic that we have is graphene. Now, in case of graphene, what we see is that you still have a hexagonal structure, right? You still have hexagonal connections, as you can see here. You still have hexagonal connections, but this time as a two-dimensional sheet, right? It's a two-dimensional sheet. Instead of being a sphere or being a cylinder or anything like that, it's a two-dimensional sheet of carbon atoms. So here you have all the hexagonal structures which are in a two-dimensional form. Now, the same graphene structures, when they stack one over the other, they make graphite. Graphite basically is essentially made of graphene, the sheets of graphene which can slip over one another, and they actually make up graphite. And that's why graphite is weak. That pencil lead, for example, you can break it very easily, because what happens is that these layers of graphene, they can slip over one another, and that's why it's easy to break graphite. Now, discovery of graphene happened in 2004, and the kinds of properties that we saw with graphene were also exceptional. It was That's why it was named as a miracle material, or sometimes also called as wonder material or miracle material. So it was named so because the kinds of properties that we saw with graphene, these were properties that we couldn't even imagine were even possible. So, for example, one of the best conductors of electricity, one of the best conductors of heat at room temperature, strongest natural substance discovered so far, thinnest compound known with just one atom thickness. So, because of all these properties in one single particle, graphene became an overnight hit. And all of a sudden, the entire world was talking about graphene. A lot of articles were written on graphene. Graphene was seen as the future of material technology, and it a lot of articles from early 2000s. I mean, 2004 the discovery happened. So if you look at those articles in 2004, 5, 6, etc., you'll see that it just looks like graphene is just going to take over the world and change the world and do everything here. Now, The discovery of graphene itself was very uh, different, very peculiar. 2004, the discovery happened, and of course, it also got a Nobel Prize in 2010. Now, in 2004, when the discovery happened, the discovery was an event which was very unique event, because the scientists who discovered graphene, they were just playing around with graphite. So they were basically using graphite, and they were just trying to peel off the surface of graphite with the help of sticky tapes, or what you call as cello tapes, etc. So with the help of a cello tape, they were just trying to peel off the surface of graphite, and while doing that, they stumbled upon graphene. That graphite, basically the layers of graphite, they actually gave away graphene. So that's why, just by playing around with the tape, they were able to win the Nobel Prize. So any one of us could have been there and gotten that Nobel Prize, but unfortunately, we were not the ones to do that, and they were the ones who got the Nobel Prize. And interestingly, the scientist who got the Nobel Prize for discovering graphene, he also is famous for getting another Nobel Prize. I don't know whether you know about this Nobel Prize. There's a Nobel Prize which is called as IG Nobel Prize. There's a Nobel Prize called as IG Nobel Prize. IG Nobel Prize stands for Ignorable Nobel Prize. And every year, ignorable Nobel Prizes are also given. And this scientist, the name of the scientist is Andre Jim. He basically has these two Nobel Prize. One, the infamous Nobel Prize, that is the Ignorable Nobel Prize. And then he also has the actual Nobel Prize, that is that he got in 2010. So in 1999, I think he first got the Ignorable Nobel Prize, because he had shown how magnetic levitation of frog was possible. That you can make the frog jump with the help of magnetic energy. This is what he got the Ignorable Nobel Prize for. So he got the IG Nobel Prize in 1999, and he got the actual Nobel Prize in 2010. The only scientist to have gotten both of the Nobel Prizes. Now, ignorable Nobel Prizes are very interesting. I mean, if you know the Nobel Prizes and how the Nobel Prizes are given, for what the Nobel Prizes are given, it will be any insignificant scientific discovery or invention. You get the Nobel Prize for that. You get the Nobel Prize for insignificant uh, uh, Nobel Prize for, I mean, for example, this year's literature Nobel Prize is very interesting. That uh, the scientists who got the Nobel Prize in literature in 2023, they got the Nobel Prize for studying the sensation that people feel when they repeat a single word many times. This was the actual paper. This was the actual research, that the sensation that people feel when they repeat the same word many times. So these kind of Nobel Prizes are given in case of ignorable Nobel Prize. And the most interesting, and this is something that relates to probably this classroom as well, to maybe some of you, not me. Uh, the Nobel Prize in Education, the Ignorable Nobel Prize in Education 2023, has been given to the scientist who carefully studied the boredom that students and teachers feel in a classroom. Right? So, carefully studying the boredom of teachers and students in a classroom is what they got the Nobel Prize, or Ignorable Nobel Prize in Education, for in 2023. So that's why all these irrelevant research is for what the Nobel Prizes or Ignorable Nobel Prizes are given. And the 1999 Ignorable Nobel Prize was actually given to the same scientist who then got then also got the Nobel Prize for discovering graphene. So it's a very unique case where a single person has both of the Nobel Prizes. Otherwise, in that scenario, there is nobody else who has this kind of an achievement, having both the Nobel Prizes. Anyway, let's move away from the trivia and try to concentrate on what we have to study. So, graphene. Now, in case of graphene, what we see is that there are a lot of different applications where graphene can be used. So as I told you that initially it looked like graphene is just going to change the world, it's going to take over the world, and everything in electronics and electrical will all be about graphene in the future. But almost 20 years later, I mean exactly 20 years later, after the discovery of graphene, we still do not see a lot of use of graphene. There are only some areas where graphene has made an impact. For example, in electronics, graphene has made an impact. Like, for example, today there are televisions which are called as OLED TVs. Have you heard of it? OLED TV. So OLED television, marketed largely by LG and Sony. These two companies, they largely uh, are the ones who have the market of OLED television. So OLED television is where they make the use of graphene. Graphene is used in making OLED TVs, and similarly, even again, foldable devices. In making of the foldable devices also, uh, graphene is useful. But wherever graphene is being used, one of the major problems with graphene is that graphene is expensive. Graphene is expensive and also difficult to synthesize in large quantity. So that is where the limitation lies, that it's not very easy to synthesize graphene, and it is expensive. So that's why you cannot just go on using it everywhere. Now, for example, let's say when we are saying that it's one of the best conductors of electricity, can we use graphene as electrical wires? Why not? We can use graphene as electrical wires, but then it will just skyrocket the cost of electricity, because right now the wires that we use are made of copper or aluminium, and these are very cheap metals, whereas graphene in that comparison will be very very costly. And that's why, in that sense, graphene practically cannot be used. Theoretically, yes, it can be used, but practically it cannot be used. So that is where we see the problem with graphene. So it can be used in various applications to enhance the strength of material, to make LED lighting more effective and long lasting. They can be used in electrical devices. They can be used in batteries and supercapacitors. They can be used as a corrosion barrier between oxygen and water diffusion. And then lastly, they can also be used in water purification. So there are a lot of applications where graphene can be used. But we expected a lot more is what we mean. Now in India, we have seen that there are certain programs now that India is also starting, and this was the news in August 2023, where a not-for-profit company called as India Graphene Engineering and Innovation Center has been set up by India. And this is basically something that will help in trying to fill the gap between the research and development and commercialization of graphene. Because although a lot of studies have been happening in graphene, we do not really see that graphene has entered Indian markets. And this is where we want graphene to start entering Indian markets. And hence, this is where this is being taken as a effect. So the name of the program is Graphene Aurora Program. Graphene Aurora Program, which was launched at Maker Village. Now, Maker Village, just remember that Maker Village is a concept that Government of India has started about innovations that can be done in technology in India. So that is where we see that in this new event that happened last year, this is where Graphene Aurora program was launched. This which can help in guiding, developing, implementing, supporting the SMEs and startups to commercialize developed graphene technologies for scale adoption. Meaning that we want it to start capturing Indian markets, start being available in their markets, and domestically they should be manufactured. So that is where we see that Graphene Aurora program becomes very, very crucial from the perspective of the future of graphene in India. Read this question. I'm launching the poll. Please mark your answers. Wonderful. 84% of you have given the correct answer. Graphene is in news frequently. Was it? What is its importance? So this question was asked in 2011 because 2010 Nobel Prize was given to graphene. So in 2011 they asked a question from here. It is a two-dimensional material and has good electrical conductivity. This is correct. And not only good, I mean, good will be an understatement. It is one of the best conductors of electricity. So this is also correct because it is two-dimensional like a sheet as we discussed. So that's why one is correct. It is one of the thinnest but strongest material tested so far. This is also correct. We discussed about this as well. It is entirely made of silicon and has high optical transparency. It is not made of silicon. It is made of carbon. So that's why this is wrong. It can be used as conducting electrodes required for touchscreen LCDs and organic LEDs. So we discussed that they can be used in OLEDs. So OLEDs are nothing but organic LEDs. So this is also correct, and correct answer that's why will be 1, 2 and 4. Now coming to another important topic, and this is the topic of quantum dots. Now in case of quantum dots, what we have seen is that uh, they also got a Nobel Prize now. The 2023 Nobel Prize has been given to the discovery of discovery and synthesis of quantum dots. Now, in this particular scenario, in case of quantum dots, understand what quantum dots are. So we have two words here: quantum dots. Quantum because they are showing quantum properties or the properties of the nanomaterial. So that's why quantum physics or quantum mechanics is largely the study of subatomic particles and at the smaller or molecular and atomic levels. So that's why the word quantum. And dot because these are spherical. All right, these are spherical material. So because they are spherical and at the same time we know that they are very small, they're spherical and they are very small. So that's why from the level that we are observing them, we can say that these are like dots for us, and that's why they have been named as quantum dots. Now the property of quantum dots is that uh, when ultraviolet light is incident on them, they show various colors. They show various colors, and the color that they show depends on the size of the quantum particle or size of the quantum dot. So size of the quantum dot will determine what kind of color it will show. If these are larger, then they will show red color. If these are smaller, then they will show blue or violet kind of color. Now the color basically is also because of the wavelength. Red has the longest wavelength, and blue or violet have the smaller wavelengths. So that's why when you have smaller sized quantum dots, they show blue color, and then when you have longer sized quantum dots, larger sized quantum dots, they show red color. So that's why color is dependent on the wavelength, and that's why these particular colors are visible. Now, in case of quantum dots, what we see is that here we are talking about very small particles, and they also have good electrical and optical properties. Now overall, when it comes to their applications, they become useful in photovoltaic devices. They become very useful in making LEDs, and here also there is something called as QLED, another kind of television that is available in the market is QLED television. And in case of QLED televisions, I mean, if you look at the advertisements, especially Samsung, Samsung is the largest manufacturer of QLED televisions. So the advertisement of these QLED televisions will tell you that these televisions can show you better contrasts, better color contrasts. So now these better color contrasts come because of quantum dots, because QLEDs will use quantum dots, and hence they will be able to reproduce colors better than normal LEDs, and that is what precisely they also advertise. So Samsung is the largest manufacturer here, plus there are other companies also which also manufacture these QLEDs. So there also in electronics we see that quantum dots are being used. Then in medical field also they can be used. We have seen that they can help in uh, the cases of cancer. They can help in cases of cancer, and we'll discuss that later also. And they can also be used as a photocatalyst. They can be used as a photocatalyst in light driven chemical conversion of water into hydrogen. So that's why they become very, very useful from the perspective of energy also, because they can be used in hydrogen production as well. So that's why these quantum dots, they do show important applications in various different fields, and hence we see a lot of advancements here. And that's why the Nobel Prize in 2023, Chemistry Nobel Prize 2023, was given to the discovery and synthesis of quantum dots. Now, overall, when we look at the various applications of nanotechnology, there are a lot of ways in which nanotechnology become useful. For example, in the field of medicine, they can be useful in drug delivery, whether gold nanoparticles or even carbon nanotubes. We discussed that carbon nanotubes can be useful in drug delivery. Similarly, in tissue engineering, we discussed that carbon nanotubes can become useful. In disease diagnostics, we see that carbon nanotubes and largely quantum dots can become useful. Then they can be used in nano carriers for eye surgery, where metal ions are used. They are used here. Then nano robots are used, that is nano sized particles are used in surgery. And in making nano flares in breast cancer treatment, here also we see that quantum dots actually are useful. So quantum dots can be used in this particular scenario. So that's why in medical field there are different ways in which we can make the use of nanotechnology. Similarly, in the field of energy, it can help us in addressing the shortage of fossil fuel, because as we discussed, if you remember the discussion with carbon nanotubes, we discussed that they have larger surface area, so that's why they can be used in energy storage also, and hence they will be able to help in making a lot of alternates. Similarly, in making nano generators for wind energy production, nanotech solar cells, where again carbon nanotubes can become very, very useful. So there also they can be used in making QLEDs. We discussed that quantum dots can be useful, and QLEDs also use less energy compared to normal LEDs. Compared to normal LEDs, QLEDs will not take as much energy as the normal LEDs take. So that's why that also is an important contribution. And nano catalyst for hydrogen production, as we discussed that quantum dots can become useful. So that's why even here carbon nanotubes become useful. Here also carbon nanotubes become useful. In all these cases, we see that these materials become crucial. Then in food and agriculture, there are silver nanoparticles which have antibacterial properties. We have nano emulsions which have antimicrobial properties. Then titanium dioxide has been seen as a very important nanoparticle. So that also helps because it also has antimicrobial properties. Then after that, we have nano bioindicators for pesticides and also for pathogens. So there are a lot of ways in which we can make the use of nanoparticles even in agriculture. Similarly, there is something called as nano urea, has been in the news also. Nano urea. So you can say in a manner nano fertilizers that we are using today. So overall, there are a lot of ways in which we see that the nanoparticles become useful in making of all these various uh, particles. So that's why they become useful in food and agriculture. So in food and agriculture, largely we see that metals especially become important, and metal ions related uh, nanoparticles can become very, very important. So that's why we can actually make the use of all these particles in food and agriculture. So in all these cases, even in making bioconjugated nanoparticles, here also metal ions are used. Here also we use metal ion nanoparticles. Then in case of the environment, so they can become useful in environment also. For example, they can be used in water filtration or water purification, where we can make the use of graphene and also make the use of carbon nanotubes. Similarly, they can help in reduction of carbon dioxide, because we can use it in various different alternate forms of energy, in solar energy, in wind energy, in even making biofuels. We want to make the use of carbon nanotubes. So that's why we see that there are all these ways in which we can help reduce carbon dioxide. Similarly, quantum dots also help in hydrogen production. So there also we see a very important advantage. So there are a lot of ways in which we see that in the environment they can be useful. Then pollution control with the help of metal oxides can be done, and then improving the air quality, air purification is possible. Here also we see that graphene becomes useful. So graphene can be used here. And in making nano sensors for detection of pathogens, here also they can be useful. So there are a lot of, again, these are just examples, and I have given you all these examples so that just in case, let's say, a question comes in the mains examination rather than in prelims, in the mains examination these kind of points can become the fodder for you to write answers. Similarly, in electronics, making nano circuits with the help of nanowires. Then the MRAMs, that is magnetic random access memory. Here also nanowires can be useful. Then the use of carbon nanotubes. The use of carbon nanotubes in electronics. We discussed that foldable displays, etc., can be made in with the help of carbon nanotubes. So in foldable displays, both carbon nanotubes and graphene both can actually become useful. Then making single electron transistors, again, nanowires can become useful in doing that, and you reducing the energy consumption, where we discussed about quantum dots and how they can be useful in making QLEDs. So in all these ways, we see that they can have their application in electronics as well. Then in the industries, in aircraft manufacturing, this is where carbon nanotubes become very essential, because they can help in making composite material. They can enhance the strength of the material while keeping them lightweight. So we see that they can enhance strength, they can enhance strength while keeping them lightweight. Then they can be used in construction industry also, and again this is basically about enhancing the strength, which actually can be done with the help of carbon nanotubes and graphene. Then they can also be used in textiles. So in textiles also carbon nanotubes become useful, and we say that these are carbon fabrics or carbon fibers. They make up carbon fibers. So carbon fibers are nothing but, you can say in a manner, carbon nanotubes or any nanomaterial made of carbon. So largely in carbon nanotubes we have these carbon fibers. So in the industries also they become useful. Then in case of space, they can reduce spacecraft waste. We discussed about that in case of carbon nanotubes. Then they can be used in making space elevators. Now this space elevator is a very unique concept, where NASA has been working on it, Japanese Space Agency has been working on it. What they are saying is that between the Earth and the International Space Station, they would make an elevator, the elevator that we use in buildings. Similarly, an elevator between the Earth station and the International Space Station is what they are saying. And they are saying that the wire here could be made of carbon nanotubes. Now, as a concept, this also has been in the news for a very long time, almost 25, 30 years. It has been in research, in the making, and obviously one of the most complex things that uh, space technology would have ever adopted. But this is also something which has been in the news on and off. I remember that almost 25 years back we would have read it in the newspaper. And this concept, when we read it in the newspaper, there was a science exhibition happening in our school, and we presented a model of space elevator based on whatever we had read in the article. We presented the model, and the jury who had come to analyze all the uh, various scientific models made by students, they came to us, they saw our model, they gave us the first prize. Neither did we understand what we were presenting, neither they understood what we were presenting. They just said that it's a very unique concept, and they gave us a first prize. So space elevator, from that time, it has been in the news, because obviously it is one of the most complex things that we have been working upon, and NASA has been trying very hard to make it a reality. Then, in making solar cells, also in making spacecraft thrusters, there are a lot of ways in which nanotechnology can become useful in space technology as well. Then, in military also, they can be useful in intelligence gathering, where especially nanowires can become useful, because they can enhance storage in a very limited space. So that is where we see that they can become useful in intelligence gathering. Then after that, in military vests, carbon nanotubes can play a big role, or even graphene can play a big role in these kind of situations, where you can make bulletproof or stab-proof clothing with the help of carbon nanotubes and graphene. And then in wound care, here this is where it becomes a crucial part of tissue engineering, and again carbon nanotubes especially become important in this particular scenario. So that's why a lot of these carbon fibers, a lot of these different kinds of nanomaterials, they see their applications in a lot of different sectors, and I've just taken some of these sectors where we can see the application of nanotechnology. Read this question, a very simple question. Please mark your answer. Hmm. So 69% of you have marked C, which is the correct answer. With reference to the use of nanotechnology in health sector, which of the following statements is or are correct? First is targeted drug delivery is made possible by nanotechnology. This is correct. We discussed about this multiple times. Nanotechnology can largely contribute to gene therapy. Yes, this is also correct. The same way in which targeted drug delivery is possible, similarly, gene therapy is also possible, because genes also can very easily be carried with the help of carbon nanotubes, and also, let's say, other nanoparticles to through the bloodstream and wherever we want them to. So it is possible. And look at the words can largely contribute to. So can largely contribute cannot be a wrong statement. I mean, just the language, even if you notice the language. So, but yes, this is possible, and we discussed that as an example that we can actually do it. Read this question. This was asked in 2022 paper. Sorry, 23 paper. In launching the poll, please mark your answers. Hmm. 64% of you have marked A, and that's the correct answer. Carbon fibers are used in the manufacturing of components used in automobiles and aircrafts. This is perfectly fine, and we discussed about this, that we can use it in aircraft manufacturing. Carbon fibers, once used, cannot be recycled. Now, this is wrong. They can be recycled, and that's what we'll try. If you remember, what I told you is that carbon fibers, or let's say carbon nanotubes, they are very strong structures, significantly strong, lightweight. So it's not that they cannot be recycled. They can be recycled, they can be reused over and over again. So there is no problem in recycling them. We would prefer recycling them because we are not able to dispose them, right? We're not able to dispose them properly. That is what I had the problem in the last statement, if you remember, that when we had discussed about the fact that they are not biodegradable. So that's why when you do not have something which is degradable, you would try to recycle them. So yes, recycling is possible, and this is, that's why this is wrong, and this should be wrong, and the correct answer should be one only, right? So let's take a break here. Let me show you the leaderboard for this part of our discussion. So this is the leaderboard for whatever discussions we have done until now. So congratulations to every one of you who have marked all the correct answers, and for everyone else, I hope that you understood what went wrong, so that next time you do not commit the same mistake, right? I have another question for you before you go into the break, and I'll launch a poll for you. Uh, we were just discussing among our teachers and the team that we have right now a purple background for your PPTs. Do you prefer this purple background, or do you want a white background? Because there were some emails and some suggestions that we got from the students, where the students say that it becomes difficult for you to take printouts if you want of all these notes. Let me just take your opinion also on this. We wanted to understand whether everybody feels that, or are there only few people who feel like that. Mark A if you want white background, and mark B if you want a purple background or the same colored background. Let me just take your opinion also on this, and I mean, with every batch we'll be doing that so that we also understand what the students feel. And there is no right and wrong answers here. I'm just randomly marking one of the answers to be correct. Now, in terms of the annotated PPTs, I'm saying that do you want annotated PPTs to be in the white background? I know the pros and cons, as many of you have written also. I understand the pros and cons, but eventually, what do you think is more useful? Hmm. So, still the preference is purple background over white. And I understand that white can become very shiny on the screen and it can take a toll here. But from the perspective of printouts is where I feel that a lot of people feel the problem, that they cannot take the printout of the annotated PPTs because of the background. So there's a trade-off there. But I think white background PPTs are already provided to you before the class also, if I'm not wrong. That there is a PDF provided to you before the class and it has white background, right? So you actually can use it. We have not taken any decision on it, just that there were certain suggestions that came in and we were thinking that what should we do, and whether we should stick to the same format or should we look at changing the format or not. Anyway, let's take a break. Let's be back at 2 o'clock and we shall continue with our discussion.